Hey everyone, and welcome to the 50th official episode of my podcast. Episode 50 is all about plant hydration. Today I'll be talking about how plants seek out and find water, how they absorb it, and how they move that water through their bodies. The basic premise of plant hydration is that the roots absorb water in the ground, and this water flows through a pressurized system of cellular pipes through the stem to the leaves, where water evaporates and leaves the plant. The leaves drying out passively brings up more water from the stem and the roots, and this encourages the roots to absorb more water. Vascular plants can do this passively, effortlessly, with no need to spend chemical energy fueling any part of the process. The details of this water flow have to do with everything from the laws of physics to the specific cellular structures that the plant has in its vascular tissue. Okay, so there's a few concepts that I need to introduce and explain first, before the rest of this will really make any sense. The first concept is one that I've talked about a lot on the podcast, like in episode 33 about plants, and in pretty much every episode in the series on ecology and biomes. This concept is transpiration, which is the formal name for the evaporation of water from the leaves and the herbaceous shoots of plants. The water inside of the leaf will evaporate into water vapor if the air outside the leaf is relatively dry, as this would create a gradient or a concentration difference between the water and the air inside and outside the leaf. Transpiration is regulated in part by structures embedded in the plant epidermis and the cuticle called stomata. The stomata are little pores created by two cylindrical guard cells pressed together in a parallel structure. When these guard cells get filled with water, they expand. And because of the specific arrangement of the structural fibers within the cell membranes, when they expand, they bend outwards in a kind of boomerang shape. When both guard cells are swollen with water and bent outward like this, they create an open space, a pore, through which gases like molecular oxygen and water vapor can leave the plant's tissues, and carbon dioxide can come into the plant's tissues. Water potential is the second concept I want to talk about, but it's one that I haven't really talked about that much in the podcast. But that's alright because it's pretty simple to understand. If you have pure water sitting still at room temperature, it pretty much has zero water potential. It's not going anywhere. This is also called zero psi because of the Greek symbol that's used for water potential. It looks like a little trident. But if you have water on a steep incline, then that will have some water potential. For example, water on an incline will have potential energy like an object that's being held up in the air. When the water runs down the incline, down the side of the hill or side of the valley or whatever it is, its potential energy turns into kinetic energy, just like the object that's being dropped from some height and falling to the ground. Water potential determines where water will move in certain circumstances. In the case of water on an incline, like a hillside or a valley, the water potential follows the path of least resistance, of decreasing water potential. In this case, the water potential up higher on the hill is high, and the water potential at the bottom of the hill is low, and so the water will flow in the direction of high to low water potential. In the case of transpiration, the water will do the same thing. It'll only evaporate out of the leaf if the air outside is drier than the air inside the leaf, and that would be the result of a gas exchange of water vapor trying to reach an equilibrium. The last concepts are solute potential and pressure potential. These are also relatively simple concepts to understand, but when applied to biological systems, they can become really diverse and complex, and they can be used in a lot of really complicated chemical cascades and chemical energy cycles. So, solute potential is basically quantifying the net movement of solutes in a solution. If you have a cell with no solutes inside its body, and you put it into a solution that does have solutes, then through diffusion, the solutes will try to get into the cell to create an equilibrium in the solute concentrations. You have the concentration inside this semi-permeable membrane, and you have another concentration of solutes outside, in the solution outside the permeable membrane. And so diffusion wants to get the solute concentration into a homeostasis, so it's equal between both bodies of fluid. This tendency for water to move from low solute to high solute concentrations, and thus simultaneously moving from high water concentration to low water concentration, to create an equilibrium, follows a gradient quantified by the solute potential. If the cell membrane is impermeable to these solutes, and the solutes can't come in to increase the solute concentration inside the cell that way, then water will leave the cell through osmosis. And as water leaves, by reducing the water concentration, it increases the solute concentration inside of the cell. And it does this to try and get solute concentration equilibrium that way. This will result in the cell shrinking and shriveling. And if it shrinks too much, it'll go past a point of no return, where its membrane has fatally collapsed. And even if you put more solutes into the collapsed cell to try and get more water in, it just can't be reinflated. If the situation was reversed, and the cell had solutes while the solution it was put into had no solutes, then the solutes inside of the cell would try to leave. They'd try to pass through the permeable membrane and establish a concentration equilibrium that way. Water would also come into the cell to try to find equilibrium that way, as the water coming in would raise the water concentration and get it close to the pure water concentration outside the cell. But doing this can sometimes cause the cell to swell like an overfilled balloon, and this will keep going until the cell pops or blows up or lyses. To a degree, plants don't have to worry about this because their cells have cell walls, and these cellulose and sometimes lignin-rich cell walls are able to contain the membrane and prevent it from expanding too far. Where an animal cell might expand until it breaks open because it's got no cell wall, a plant cell will expand until it presses against the inside of the cell wall. And this pressure will increase, and it can get to pretty high pressures, but that cell wall isn't going to give way, and it's going to prevent the membrane itself from swelling too much so that it destroys itself. That pressure that's created from the water swelling and pushing against the cell walls, that's where pressure potential gets involved. When the plant's cell membrane is not fully expanded, it has what's called a low turgor pressure. When it has enough solutes such that the water coming in fills it up, it'll have a higher turgor pressure. The membrane will be more full and more taut, like a full water balloon. When the membrane gets so full of water that it pushes up against the cell wall and the cell wall pushes back, this is called cell wall pressure. Plant cells that are turgid are well hydrated. Their cells are stiff because they're experiencing more wall pressure. This is why well-hydrated plants look healthy and firm and strong. Well, if you look at a dehydrated plant, one that hasn't had water in a few days and it has low internal turgor pressure, you'll see that its branches, its limbs, its leaves are limp and flimsy and soft. This is the macroscopic physiological effect of pressure potential, of the turgidity in the plant cells. Okay, so all of these concepts that I just covered, they all play a role in plant hydration. The roots first have to find water somewhere in the soil, and when they do find water, they can only absorb it effectively if the water potential in the roots is lower than the water potential in the soil. Because water flows from high potential to low potential, the high water potential that's typically found in rich soils that'll hold on to a lot of water, it allows that water to move into the roots, which have a relatively low internal water potential. This isn't the case with silt-rich water, as the huge amounts of solutes in the water in the form of sediment drastically lowers the water potential. Really dry soils are also difficult for plants to handle, because in really dry soils, water will first want to stick to the dry mineral and soil particles before it'll want to get absorbed into a plant's tissue. The lack of soil saturation means that water is retained through surface tension on these soil particles. Because that surface tension is kind of difficult to break, the roots really struggle to absorb that water. Only when the soil has been decently saturated with water will the water potential rise, and the water will then begin to flow into the root tissue. In other cases, where the external water potential is really low, lower even than the water potential in the roots, the plants can be in a lot of danger because water is going to flow from high to low water potential. And if the roots have a higher water potential in the soil around them, the soil will actually start to suck water out of the plants and back into the ground. If you're a plant living somewhere that's really dry and the soil all around you is really dry, it's pretty much a death sentence when the soil starts taking water back out of your body. Above ground, the plant's shoot is exposed to the air. Water also exists in the air as vapor, and even this has a water potential that plays a role in plant hydration. When it's warm and dry outside, the water inside the plant's leaves exists at a much higher water potential than the air outside. The inside of the leaf is a hydrated, humid place, so air that isn't humid will necessarily have a lower water potential. Because water vapor will flow from high to low water potential, just like liquid water, this will take the vapor out of the leaf and into the dry air, into the external environment. When it's humid outside, on the other hand, the water potential of the air is closer to that inside the leaf, and so the rate of transpiration isn't as fast. Transpiration will set up a water potential gradient along the height of the plant, along its vertical axis. In the soil, where all of the free-range water is, the water potential is really high. This means that there's a lot of water there. There's a lot of water in the ground. It's wet. It just so happens that the plant grows roots, and these roots stick into the ground. They grow into the ground and penetrate deep into the soil. And because of the slightly lower water potential in the roots, they absorb water out of the ground and bring it into the plant's body. The leaves of the plant have a water potential that's much lower than the roots, and the air outside the leaves has an even lower water potential. The real magic happens when water moves between these two realms, when water and sugar are passively brought up from the roots to the leaves, through the plant's vasculature, moving up against gravity, but down the water potential gradient. At the root, the water is absorbed, and it moves through the cells of the cortex. Water can move through the phospholipids in the cell membranes. It can be channeled through the cytoplasm of the cells, where all of them are connected through protein channels and pores and stuff like that. And water can even flow through the pores of the cell walls. All of this water is moving to the center core of the root, to the vascular tissue, and all the cortex cells the water moves through act like filters that moderate the incoming solutes, so that the water that makes it to the vascular system has a controlled solute concentration, and this won't damage the plant by throwing its osmotic balance out of whack. Another way that plants regulate this water balance is with an evolved layer called the Casparian strip. There's a layer of endodermal cells, and these endodermal cells line the vascular bundles, or they line the vascular tissue, and they separate it from the cortex. They secrete a waxy substance called suberin into their cell walls, and this suberin will clog all of the little pores, and they'll clog all of the holes between the cell walls of individual cells. When all of this suberin is excreted into a sheet that connects all the endodermal cells, it forms a watertight seal. This also prevents water from flowing through the cell walls into the vasculature, and so it kind of forces any water flowing into the vasculature to have to go through the cells, through cell membranes. In this way, it's kind of like a mandatory filtration service. Once water has passed the Casparian strip, it flows into the phloem and the xylem, and these are the two major vasculatory cell types that enable water transport. In small plants, a phenomenon called root pressure might be sufficient to push water up and out of the plant. At night, when there's no photosynthesis and no open stomata, water flow through the plant tissue is diminished. But keep in mind that the roots don't need sunlight to accumulate nutrients, and so they pretty much always accumulate nutrients, and as they do, they fill their cells with solutes. This causes water to continually flow into the root cells, and they expand with water until they reach a decently high internal pressure. This water pressure at the roots, the root pressure, will build up until it starts to push water up into the stem and the leaves. This results in a process called guttation, wherein root pressure pushed water up and out of the roots, through the plant and out of its leaves. If you look at plants like grass and small bushes early in the morning, you might see little water droplets on the end of their leaves. This is the guttation caused by root pressure pushing water up through the leaves. The physical properties of water, called adhesion, cohesion, and surface tension, also play a really important role in the plant's ability to passively move water up through its body. Adhesion is a quality created by the hydrogen bonds that transiently form as different molecules come into contact. If you look at how water acts when you have it in a glass cup, you'll see that at the very edge of the cup, the water rises up in a slope as it approaches the glass. This is the adhesion of billions of hydrogen bonds, pulling the little rim of water up onto the glass against the force of gravity. This same phenomenon occurs in the cellular pipes of a vascular plant. The cellular vessels and capillary tubes interact with water through adhesion. Cohesion is the attraction between similar molecules. It's a lot like the transient hydrogen bonds that form between polar molecules. When adhesion pulls some water up the sides of the container, the force of cohesion pulls up the rest of the water column below the elevated surface. These forces in a tube together create a meniscus at the interface of air and water, which is when the water at the rim is held up by adhesion and filled in with water that's pulled up through cohesion, while gravity pulls down the water in the middle, and it makes a little concave depression on the surface of the liquid. Surface tension is the force that pulls across the surface to minimize its surface area. If you have this concave shape, it's going to have a slightly greater surface area than if you just had a perfectly flat circular cross-section or a circular interface between water and air. Thus, surface tension and surface area fights against gravity to lift up the center of the meniscus. When you're talking about water flow through cylinders like those you'd use in a typical science lab, the effects are plainly visible. You can look at them and you can see the meniscus, you can see the effects of adhesion and cohesion, and you can see surface tension. These qualities of water don't really help the water get that much higher up the tube. You just have that little bit of adhesion kind of pulling it up slightly on the side. In the plant's vasculature, however, the water is being moved up extremely tiny pipes, cellular pipes so small that they're literally composed of single-file columns of cells. In these extremely confined tubes, these three properties of water work together to fight against gravity, and they allow the plant to lift the water tens and even hundreds of feet into the air. The adhesion first pulls up water against the edges of the vessel elements. When the night comes to an end and the sun emerges, it'll heat up the upper portion of the water, and this will transpire away into water vapor, and this will create a deep meniscus. The space left open through the meniscus will get filled as cohesion brings up the water column. Surface tension in such a small space pulls the center of the meniscus up, and it also raises the water level. So in these structures, water kind of naturally, passively just gets pulled upwards. Although understand that water potential, the internal pressure, and the transpiration through the leaves are really critical drivers of this process. But as long as there's sunlight, the process really does kind of sustain itself in a form of perpetual tension. As long as the leaves have a low water potential and the air outside has an even lower water potential, the tension at the water-air interface will passively pull water up through the plant in a continuous stream. This is a solar-powered process, as the drying out of the leaves perpetuates the tension. Through cohesion of the water molecules in the continuous column, this tension reaches all the way from the leaves to the roots. There was a really important evolutionary event, millions of years ago, that enabled vascular plants to grow really tall. I mean, even though there's this neat little cellular trick that vascular plants have to bring water up against the force of gravity, that water still has a lot of mass. Water is heavy, and a continuous stream of water running through a plant and out its branches is a lot of water. It adds up. Herbaceous plants are thus limited in size because their soft tissue just can't support the weight of too much water. Even when they're fully turgid and stiff, too much water is too much water and the plant can hurt itself or simply collapse. But millions of years ago, a molecule called lignin was evolved and it emerged in the cell walls of plants. These plants today incorporate that lignin into their secondary cell walls, and this creates hard, dense, woody plant tissue. This evolution of much greater structural support allowed plants to hold on to more water, and soon there were species of woody shrub and woody tree that reached really high into the sky because they had the durable, sturdy tissue that allowed them to do it, that could hold up that water weight. This continuous flow of water across the water potential gradient from roots to shoots can only be upheld as long as there's a continuous supply of water. When plants start to feel water stress, they can't keep up this continual flow, and the solute potential in their cells and their tissues will drop until the whole plant begins to lose turgor pressure and wilts. To prevent total collapse of the water column, plants will often close their stomata to lower the rate of transpiration. But this has a secondary effect that interferes with photosynthesis. With closed stomata, there can be no gas exchange. And with no gas exchange, the plant can't take in the carbon dioxide that it needs to grow, and it can't exhale its waste oxygen back out into the atmosphere. This relationship between water conservation mechanisms and the gas exchange necessary for photosynthesis is called the photosynthesis-transpiration compromise. Plants in various habitats have to manage this compromise in such a way so that they don't run out of water or totally shut down their photosynthesis. So then, that raises the question, how do plants prevent water loss and maintain a continuous flow of water, especially when they live in dry climates with dry soils? Some plants like spruce trees and pine trees have needles like leaves. These needles have a smaller surface area, and so there's less surface area available total for transpiration. Limiting the surface area exposed to the air limits transpiration. This is form follows function, and so this helps the evergreen trees conserve water. Another common feature on all sorts of plants is a cuticle, which I've also mentioned numerous times in the podcast. The cuticle is a waxy layer secreted by epidermal cells which creates a waterproof layer between the outer cells and the air. This protects them from undue water loss. I've also mentioned and explained stomata. There's an adaptation to dry air, one that I think is particularly fascinating, where the stomata are clustered on the underside of the leaves, away from the sun, and they're clustered in these little bowl-shaped depressions or cavities in the underside of the leaf's tissue. These little pockets or craters running along the underside of the leaves have little hairs called trichomes ringing the opening. These little trichome hairs protect the pocket, and they create a tiny microclimate of still air. Because the stomata are exposed to this semi-controlled microclimate, their rate of transpiration is suppressed compared to the stomata that are open directly to the dry, windy atmosphere. The flow of air through the stomata, it can't be 100% closed off though, because carbon dioxide needs to flow into the plant so that it can conduct photosynthesis and so that it can produce carbohydrates like starch and cellulose. Some plants utilize a hyper-efficient form of photosynthesis called C4 photosynthesis, which prioritizes the accumulation of carbon dioxide into things called bundle sheath cells. And these exist deep inside the leafy tissue, away from any stomata that might leak the carbon dioxide back out into the atmosphere. Other plants use a biochemical pathway called CAM, C-A-M, which I've mentioned once before on the podcast. CAM stands for crassulation acid metabolism. The CAM plants will only open their stomata at night when it's cool and dark and transpiration is slow. The carbon dioxide accumulated during the night is chemically associated with various acids, hence the crassulation acid metabolism. And during the day, when photosynthesis starts up again, the acids release this carbon back into the Kelvin cycle, where it gets processed into sugars. This sugar is dissolved in the water and transported through the vascular tissue as well, as the sugars produced in the leaves need to go all over the plant's body, so that its roots, its stems, its branches, and its leaves can all grow. The movement of sugar through the plant can be understood by looking at it as a system of sources and sinks, or basically areas where sugar is produced, sources, and areas where sugar goes to be stored or goes to be used or consumed, and these would be sugar sinks. At the beginning of the growing season, as the plant is awakening from its winter dormancy or returning to life after a dry season nap, any sugar that's stored in its tubers will get tapped to fuel the initial growth of the new season. In this case, the tubers are sources, and the freshly growing tissue, like all of the new leaves and the growing branches, are the sugar sinks. During the growing season itself, the photosynthetic tissue in the stem and in all of the leaves become sugar sources. With the ample sunlight during the summer or the wet season, these tissues are photosynthesizing a ton of sugars, far more sugar than the leaves or the stem need on their own for their own growth and sustenance. The excess sugar from all of this photosynthesis will get shuttled around to the structures that need it, but aren't themselves producing sugar. They're sugar sinks, not sources, which includes structures like the apical meristems, where all of the new cellular division is happening, areas like the storage structures, like tubers that exist in the roots and the underground shoots, and the sugar-rich reproductive structures, like fruit and seeds. Where water flows through both the phloem and the xylem, sugar only flows through the phloem. Where water moves from high water potential in the roots to low water potential in the leaves, sugar moves from the high solute concentration in the sugar sources to the low solute concentration in the sugar sinks. The phloem that connects sources and sinks is often dependent on the location of each. For example, leaves on the lower half of the tree will typically send their sugars down to the roots, while leaves on the upper half of the tree will send their sugars to the new leaves growing beside them in the apical meristem in the canopy. Further still, specific leaves will only feed sugars to the leaves and the roots nearest them, or on their side of the plant. This location-specific distribution of sugar is the result of the structure of the phloem. The phloem is composed of two types of cells, called sieve-tube elements and companion cells. The sieve-tube elements are really simple structures, to the point that they're hardly even cells. They have cell walls, which gives them their distinctive shape and their strength, and they're connected to other sieve-tube elements above and below them through structures called sieve plates, which are like flat plates perforated with holes that provide a direct connection between the membranes and the cytoplasm of the adjacent sieve-tube elements. Besides this, sieve-tube elements don't really have any organelles. They don't even have nuclei. For all intents and purposes, they're just empty tubes with an inner lining of a semi-permeable membrane. The companion cells, on the other hand, are wrapped around these sieve-tube elements, and unlike their tube friends, the companion cells are packed with organelles. The companion cells are so named because they compensate for the sieve-tube elements by packing extra mitochondria, extra ribosomes, and extra organelles, extra nuclei, and all sorts of things to help support and grow the sieve tubes. The sugar fills the sieve tubes along with water from the xylem, and this creates a sugary sap that moves through the plant, with independent phloem connections between specific sugar sources and sugar sinks. This sugary solution is propelled not by transpiration, but by differences in turgor pressure between cells at the source and cells at the sink. Also, unlike the flow of water up a tree, sugar transport does require an energy input. It's not a passive process. So at the sugar source, sugars like sucrose are being created, and they're diffusing across the companion cells into the sieve-tube elements. This increases the solute concentration inside the sieve-tube elements, and it pulls water out of the xylem. The water flows into the sieve-tube elements in an attempt to lower the solute concentration and establish homeostasis again. But this just creates a strong turgor pressure in the cells of the tissue at the sugar source. At the sink, the opposite is happening. The tissue is actively pumping or passively sucking sugar out of the sieve-tube elements and into the growing tissue. This pulls solutes out of the sugar sap in a process called phloem unloading, and it lowers the solute concentration of the phloem fluid. A lower solute concentration will encourage water to leave the sieve-tube elements near the sinks, and this puts water back into the xylem. So basically, phloem is under pressure, and this pressure propagates the movement of sugar sap throughout the plant's body. But to begin this process and create the gradient in the first place, the plant has to move sugar from the sources to the phloem. During the middle of a summer day, when a leaf is photosynthesizing at its maximal rate, it's producing a lot of sugar. In this circumstance, the cells in the source tissue are packed with sugar, and it flows out into the sieve-tube elements through simple diffusion across the cytoplasm and through the pores between cells. But when the leaves and the other sugar sources aren't operating at full capacity, they can't necessarily rely on simple diffusion. The plant has to actively pump sugars out of the sinks and into the sieve-tube elements, so much so that this pumping raises the solute concentration of the sieve-tube cells and keeps it high, so as to continually bring in water from the xylem and establish that high-pressure end of the gradient. This active pumping that establishes that pressure gradient is itself established through a system of protein pumps and channels. First, a protein pump in the membrane of a companion cell will consume ATP to pump hydrogen ions, or protons, out of the cell. So this process burns chemical energy to increase the ion concentration of positively charged protons immediately outside of the companion cell. And this establishes an electrochemical gradient, wherein the protons want to flow with the concentration gradient back into the cell. This gradient of proton flow is exploited by other protein complexes called symporters to shuttle other molecules into the cell as well, but against their concentration gradient. These symporters pretty much tap into the energy that can be released when you let a proton come back in, along with its concentration gradient. You harvest that energy, and it'll allow that protein to pull in a different ion or a different molecule against its concentration gradient. This might seem a little confusing, so let me try to simplify it a little bit. You have a membrane with a symporter protein embedded in it. Outside of this cellular membrane, there's a lot of protons, but only a little bit of sugar. Inside of the cell, there's a lot of sugar, but only a few protons. Thus, the proton concentration gradient, going from high to low, has the protons wanting to move into the cell. But the sugar concentration gradient runs the opposite way. If we let this process go on just with simple diffusion, the sugar would try to reach equilibrium by leaving the cell. This is a problem when you want to load sugar into the cell so that you can create a pressure gradient in the phloem. The symporter protein will thus tap the energy in the proton gradient to bring a sugar molecule into the cell against its concentration gradient. Because these symporters bring sugar into the companion cells, the sugar has a direct route into the sieve-tube elements. The sugar has now been loaded into the phloem, and it's traveling through the vasculature to a sugar sink. At the sink, the phloem needs to be unloaded of its sugar content. When the sink is rapidly growing tissue, like new leaves that are using the carbon in cellular respiration and for the physical construction of biomolecules like proteins and carbohydrates, the sugar is unloaded through simple diffusion. The sugar-hungry leaf tissue sucks up all the sugar it can get from the phloem, and it produces all the new tissue and the new biomolecules that it needs. Because this kind of sink is hungry for sugar, the unloading process is passive. It requires zero energy input. The process works differently at sugar sinks where there's no growth, but only sugar being loaded up into storage. These storage points create areas of highly concentrated sugar. Because of its high concentration, there's a strong concentration gradient wanting to take that sugar out of storage and out of the cells. To prevent this, to keep the sugar in storage and prevent it from leaking back out through simple diffusion, the plants will store sugar in a large empty organelle called a vacuole. The membrane of this vacuole is called a tonoplast, and it contains two specialized pumps. The first pump is used to move protons against their concentration gradient into the vacuole. Then the second pump comes in, and it uses these protons wanting to move back out of the vacuole along their concentration gradient, and they use that energy to pump a sugar molecule against its concentration gradient into the vacuole. This is kind of interesting, because by containing the sugar inside of a closed subcompartment within itself, the rest of the cell has relatively low sugar concentration, and thus it has a low turgor pressure, and this encourages the passive flow of sugar from the phloem into the cells at the sugar sink. By isolating the sugar, the zinc cells maintain a low turgor pressure, and thus they maintain the whole pressure gradient in the entirety of the phloem that supports this perpetual movement of sugar. Alright, well, that's about all that I have for you for plant hydration. This was a pretty fun episode. I enjoyed researching it and writing about it. I think plants are really fascinating, and the way that they move water in a way that almost seems to exploit the laws of physics is really cool. It's really interesting. I also hope that you thought this was a fun episode as well. I hope you learned something cool. I hope you learned something that you thought was really fascinating. I hope I was able to shed some light on how plants grow and hydrate themselves, and how they move water against the force of gravity to seemingly impossible heights. If you found this video particularly enjoyable or educational, consider sharing it with your friends or your classmates or your teachers. Hell, consider sharing the whole podcast. Tell your friends and your classmates and your teachers about this kick-ass biology podcast. Help me get the word out there. Stay tuned for the next episode, where I'll explore plant nutrition. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.